So let's start this next discussion with a new term, that is ADME, or absorption, distribution, metabolism, and excretion of drugs. These characteristics taken together define the molecule's drug disposition. We've talked already about in previous lectures about two different classes of protein that are involved in determining a drug's disposition, namely the xenobiotic metabolizing enzymes. Primarily, we focused on the cytochromes P450, but we talked about some other phase 2 enzymes such as UGT that are involved in attacking hydrophilic functional groups onto a drug molecule to facilitate excretion. We also talked about the receptor molecules that are involved in sensing the presence of xenobiotics and communicating that presence to the cell and affecting transcriptional change as well as other kinds of changes. But there's a third class of molecules that we'll talk about today that participate in determining drug disposition, and those are the drug transporters. These molecules are involved in the uptake of drugs, that is, the translocation of drugs into cells, and the efflux of drugs, and that is the export of cells away from the export of drugs away from the cytoplasm and into waste. What I'll say about uptake first is that these molecules tend to function as symporters or antiporters, and that is to say that they utilize a concentration gradient of another molecule, perhaps a small ion, sodium or carbonate or protons, and use that concentration gradient and the re-equilibration of that small molecule across the concentration gradient to deliver the drug to one side of the cell membrane or another. Those can either go in the same direction across the membrane as a symporter or in an antiporter. Then as one solute is taken in one direction, the other is carried in the opposite direction. That's in stark contrast to efflux transporters, which require the hydrolysis of ATP to deliver the molecule and move that molecule against a chemical gradient, so uphill energetically, as it were. As we'll see, these molecules, these transporters, are localized in a variety of tissues, but principally in the tissues that might make most sense in terms of, again, regulating the pharmacokinetics of xenobiotics, the intestines, the liver, the kidney, and interestingly as well, perhaps the brain. And this might become more important as the semester rolls along, thinking about how drugs get across the blood-brain barrier, and that is mediated in no small part by drug transporter molecules. Okay, so what I've given you here is a list of major transporter classes as defined in the Callow chapter that I assigned for reading this week. I'm giving you these sort of common names or sort of simplified nomenclature for describing the kinds of transporters that we're interested in. That article, as well as other review articles that I gave you to read this week, use different kinds of nomenclature, and I'll touch on those as we go. Suffice it to say that there's a wide range of transporter molecules that bear no sequence or structural similarity to one another, and yet are all categorized in this superfamily of transporter molecules. There's been some effort to make that nomenclature more intelligible, and I'll hopefully be able to show that to you and help you see that it is slightly more intelligible than this one. So again, here they are, as outlined by Callow et al. Organic anion transporting polypeptides. These are obviously transporters of organic anions, right? Negatively charged species. These are as opposed to the organic anion transporters, or OATs. So same idea, organic anions being transported across the membrane, but these two classes are separated from one another because they have very little or no sequence identity to one another, regardless of the fact that they do the same thing. Now, same thing, that's not quite fair. In terms of the xenobiotics, these two classes transport. One transports lipophilic organic ions, and the other, the organic anion transporter, actually delivers hydrophilic anions. So it's it's complicated, but that's a general way that we might be able to break them down. I say it's complicated, and here under the OATPs, you see that sometimes xenobiotics that the OATPs transport are actually organic cations as well. So it's not as cut and dry as these names might lead us to expect. But that should also be no surprise given the diversity of molecules that these proteins are charged with moving across membranes. I've also shown here some endogenous targets for these molecules. It's important to remember that many of these molecules are operating within the parameters of normal metabolism, that is not being challenged by xenobiotics. So endogenous ligands for the OATPs would be bilirubin, prostaglandins, bile acids, steroids. And we can also look at the cation transporter, OCTs, and in which we're transmitting or transporting monoamine neurotransmitters endogenously. So that's interesting to think about, that those neurotransmitters are frequently going to be cationic and, of course, need to be shuttled back and forth across membranes regularly. In terms of xenobiotics, the OCTs transmit antiarrhythmic channel blockers. Now, we discussed these last week when I was showing you examples of the examples of lidocaine and mexiletine. Those are channel blockers that are known to be transmitted by the OCTs. Peptide transporter, PepT, yet another family involved in the uptake of peptidomimetics. We'll discuss some of those as the semester moves along in terms of the development of new pharmaceuticals. And quite separate from these anion transporters is a class of proteins referred to generally as multidrug resistance proteins. These are characterized by a particular structural feature, that is the ATP binding cassette, that identifies them as efflux transporters, that is, shunting xenobiotics out of the cell and into the waste. There's a whole slew of these as well. Multidrug resistance related protein, or MRPs, a variety of MDR proteins such as P-glycoprotein we'll discuss in detail next week, and we'll also look at other molecules such as the breast cancer resistance protein, BCRP, or the biosalt export pump. Again, all characterized by the presence of this ATP binding cassette that drives that uphill movement of solutes across membranes. Now, I said that the transporter nomenclature was complex and confusing, and I don't think there's a good way to get around that. Various nomenclatures are still in use, although there has been a move to simplify those, and as I'll try to explain. But now we refer to these as the SLCs, solute carriers, right? SLC, 300 genes, again, grouped into families not based on any sequence relationship, but by virtue of the fact that they all are drug transporter molecules, or transporter molecules of some type. So divided into 55 families, each of which having a different function and a different structure and sequence from their partners, from their neighbors in the family. And these are mostly endogenous transporters. So there are here I'm showing up to 22 different SLC families, and mostly these are shunting endogenous molecules, such as in the SLC2s, or the glucose transporters. Those are transporting carbohydrates, obviously, across the cell membrane. I've just shown you PepT and OATP and OCT's new family nomenclature, and this is, you can see indicated by the italics that these are actually referring to the genes. So SLC15 being the PepTs, OATPs being the SLC21s, but those are more commonly referred to as the SLCOs now. The SLCOs themselves divided up into 6 different groups, and of those groups, SLCO1 split into 4 different genes, SLCO1A2 representing OATPA, which we'll talk about at some length, and then SLCO1B1 representing OATP OATPB, now referred to in terms of the protein as OATB1B1. So just so you're clear on that, the OATP1B1 refers to the protein, whereas SLCO1B1 refers to the gene. Anyway, I'm only showing you this to give you a sense of the... Now in this slide I've just shown how different transporter molecules participate in different phases of xenobiotic metabolism. And I think it's fairly straightforward and easy to interpret. We see that the organic anion transporters and the organic cation transporters are playing a role in the import of the drug molecule from the blood plasma to the hepatocyte, the liver tissue, for degradation. I should note that these molecules are not strictly localized to the hepatocyte, but it's a logical way to represent how they're functioning. So we're delivering the drug molecules to the hepatocyte for phase 1 metabolism from the system, and then the ATP binding cassette proteins, such shown here on the bottom left, MRP3 and 4, as well on the right as BCRP, the breast cancer resistance protein, and then the multi-drug resistance proteins, and there's the bile salt export pump, all pumping these molecules out of the hepatocyte after degradation or after modification in phase 2 metabolism. So now they're ready for excretion. So these are the different roles that the transporters are playing in terms of xenobiotic metabolism. Let's talk briefly about localization. Again, I think the main thing I'd like you to take away from this kind of figure is simply that mostly the well-understood transporter molecules are playing a role in tissues that are sensible, right? Uptake and efflux, the intestine perhaps being an important place for, or a logical place for transporters to be positioned, as well as the liver and kidney. We see in OATPA that this molecule is localized to neural tissue. Now, this is really interesting in terms of thinking of its potential role in the uptake of molecules across the blood-brain barrier. But this is also a bit of a historical artifact in terms of where this molecule was originally identified. I think its localization is thought to be a little bit more complicated at this point. All these other organic anion transporter proteins, I've given you some various excuse me, I've given you some various locations for them. It's interesting to note that OATPI has only been identified as a gene, and the protein product of that gene hasn't been observed anywhere. Okay, a little bit more complexity here in terms of localization and function. So, of course, just as with the cytochromes, we have broad substrate specificity in these transporters. So we bind and transport quite a wide variety of molecules of differing structural features. In terms of the structural features of the OATPs themselves, however, we're talking about 12 transmembrane helical proteins. I'll say, because we're going to look at some models in a few minutes, that it's easy to identify transmembrane helix proteins just by their obvious patterns of hydrophobicity and hydrophilicity in terms of side chain chemistry. It's much harder to decide how those 12 identified helices might pack together into a detailed three-dimensional arrangement. Very few transmembrane protein crystal structures have been determined, but that doesn't stop people from trying to build models, and as we'll see. Okay, now these OATPs are involved in carrier-mediated uptake, the carriers in this case being frequently sodium, proton, and carbonate ion, other glutathione adducts, and that's another interesting issue since we've seen glutathione previously as adducts of glutathione as phase 2 modifications of drug molecules. In any case, let's move from left to right from the enterocyte, so it's logical to think of uptake from the enterocyte into the enterocyte from the gut lumen, and OATB actually plays a role in uptake in this case. So those heavy arrows indicating the flow of the drug molecule. So we've absorbed the molecule from the lumen of the gut, and now we're ready to send it off to the blood, perhaps, so that it can reach the hepatocyte. And if we move on to the hepatocyte, we see that the OATP B, C, and 8 are all known to localize in the hepatocyte and pump drug molecules into the liver cells from the blood. Finally, let's look back at OATPA again. Here we've identified OATPA as being present in the proximal renal tubule of the kidneys, and interestingly, this molecule was early on identified as a reuptake transporter. So we're bringing the drug molecule back from the urine into the kidney, which is an interesting wrinkle in terms of understanding a drug's pharmacokinetics if there's reuptake from the waste stream. Now, I'm showing you this slide because I really like to look at phylogenetic maps, but really, again, within a family of the solute transporter molecules, we see decent conservation, and that means structural conservation as well. We see a divergence between families, or within this family of starting about 200 million years ago. You probably know that's the Mesozoic, and in that era, we saw a fairly large speciation explosion taking place. So we see that across organisms, certainly we see, within the mammals, we see sequence relationships between the organic ion transporting proteins, and with the most divergent species, of course, being between the mammals and the C. elegans, for example. Let's see. One thing that we can say when we look at these sequence relationships is that we might take sequence identity or sequence relationships to indicate some conservation of functionally relevant structures, perhaps binding structures. So this is just maybe part of the justification for doing the following, which is building structural models of organic ion transporters and other transporter molecules just based on similarity, perceived similarity to other better understood transmembrane proteins. In any case, first of all, modeling studies identify 12 transmembrane helices. As I told you, that's fairly straightforward to determine based on sequence gazing. But further model building of mashing those helices all together within the context of a membrane results in something like 900 cubic angstrom relatively open, relatively hydrophilic pocket. Now, a model has emerged, and that's outlined in this Journal of Membrane Biology paper that I have not posted for you, but if you're interested in it, I can give it to you. In any case, the model they suggest is that there's a lever arm sort of motion that happens, only one side being exposed to the cytoplasm or the intracellular, the cytoplasm or the extracellular matrix at any given time. And the binding itself helps drive the transport process by inducing maybe a conformational change. Not hard to imagine that could take place, and then one side closes and the other side opens and releases. Remember that this is happening concomitantly with not only the drug molecule but also the co-solute, perhaps sodium or bicarbonate ion. In any case, these maps are very crude, and therefore any interpretation of single nucleotide polymorphisms, which of course is what we're really interested in this discussion, can only be very mapped in a very rudimentary way and in a very model dependent way. On the bottom here, let's see, I've just labeled this intracellular phase. So now we're looking down at the membrane, and what we see is the closed side. So you see that these helices have effectively sealed off the extracellular environment, and then when drug binds, perhaps we'll see a change in that conformation. Or when drug is released from the other side, perhaps we'll see a change in the conformation. So let's see how those polymorphisms might actually map. Before we do that, there's one other thing to